Good afternoon to all the EAPCET aspirants. We are in the last chapter of the unit Plant Physiology, that is plant growth and development. First, let us go through the contents of this chapter, like what are the concepts that we have to cover from this chapter. First is growth. About growth, which is the characteristic feature of living, we have already learnt in the diversity of living world, but here we have to address the growth which is open form of growth in plants. Fine, right? And then we have to go through, is growth measurable and in what ways you can measure the growth of plant. Growth measurable. Fine. And then go on the highlighted regions of the plant growth, like meristematic zone, elongation zone and differentiation zone. Zones of growth. Followed by differentiation, dedifferentiation and redifferentiation concepts. Dedifferentiation and redifferentiation. And then comes the concept of development. Developmental patterns will be different in different plants, where we will learn this under the concept of plasticity. One major phenomena of plasticity will be heterophily. Heterophily, we have two types of heterophily. One is based on the phases of growth and second is based on the habitat heterophily, on the environment where the organism lives or grows on. Fun, right? And then after the development, that is the concept of plasticity and heterophily, we will go through plant growth regulators. Plant growth regulators. We have two types of plant growth regulators. One is plant growth promoters and second will be plant growth inhibitors. Plant growth promoters will be auxin, cytokinin and gibberellins, and plant growth inhibitors will be abscisic acid, whereas ethylene will have wide range of activities on the plant. That is why ethylene is the only phytohormone which is widely used for agriculture purposes. So, ethylene will have both promoting effect and inhibitory effect, and the fifth hormone, phytohormone, we are going to learn is ethylene. So, these are the concepts that we are going to uncover from this chapter, plant growth and development. I hope this is clear to you, and then we will move on to first and foremost thing is seed germination. Okay, seed germination, we can find two patterns of seed germination. One is epigeal seed germination and second one will be hypogeal type of seed germination. What is epigeal seed germination? You see here, epigeal seed germination is observed in those seeds wherein hypocotyle emerges first and cotyledons will rise above the soil. Cotyledons which are addressed as embryonic leaves. Embryonic leaves, which will provide the required nourishment during the time of seed germination. So, here if you see in epigeal type of seed germination, what happens, ma? Hypogel grows first, hypocotyle grows first and cotyledons will emerge out of the soil. Cotyledons emerge out of the soil. Post this, hypogeal seed germination, wherein epicotyle grows first here. Can you see epicotyle, which is the future shoot? This epicotyle will grow first and cotyledons will remain underground itself. Okay, Here epicotyle grows first, and cotyledons remain in ground itself. Cotyledons remain underground. Fine, right? Any doubts anywhere, ma? Fine, right? Any doubts anywhere? Now, if you have to encounter the examples of hypogeal and epigeal germination, hypogeal germination you can take the examples of groundnut, which is Arachis hypogea, and epigeal germination you can consider the examples of bean family, bean, castor and onions. These will be the examples for epigeal seed germination. Clear, right, ma? Any doubts anywhere? And then after types of seed germination, one more major type that you have to address is viviparous type of seed germination, which is widely observed in those plants which grow in halophilic conditions. Plants that grow in stress, any kind of stress like salt stress, water stress, drought stress, usually they will emit viviparous type of germination. Best example is mangroves, okay. Those which grow in, plants grow in halophilic conditions. Which grow close to the seed shores. There seed is unable to germinate in such environmental conditions. So, to escape the seed dormancy, seeds when they remain in the plant itself, they start germinating, and these birds landing onto the substratum, they will grow into a complete plant. Okay, seed germination occurs when it is on the parent plant itself, that is viviparous type of seed germination. The seed germination is happening on the parent plant because the environmental conditions, the halophilic conditions there will not allow the seed to germinate on the ground. Fine, right? That is the reason they are germinating over the parent plant itself. Best examples include mangroves, fine. Mangroves are the best examples for viviparous type of seed germination. And then look into the growth pattern. Growth, which is the characteristic feature in plants, you can see both increase in cell number and increase in girth of the plant size. And in plants, the growth type is addressed as open form of growth. Open form of growth. This is because of lateral meristems or secondary meristems. Okay, lateral meristems or secondary meristems. Here you see first shoot apical meristem and root apical meristem, which are at the terminal regions. They focus only on the cell division at the terminal regions, both at RAM and at SAM. At the terminal regions, cell number will be increased, thereby the length of the plant will be increased. But if you consider vascular cambium and cork cambium, which will increase the girth of the plant, okay. Vascular cambium or cork cambium, it will increase the girth of the plant, okay. The girth or the diameter of the plant will be increased. So, that is addressed as what? Open form of growth. and these are known as lateral meristems because they appear in the later part of the plant life and they are involved in increasing the girth of the plant. So, those secondary meristems include both vascular cambium and cork cambium. Clear till here? And then, now is growth measurable? Yes, growth is measurable on various parameters in the plant species. If you see estimation of growth, increase in dry weight or fresh weight, that is how you will see the increase of growth in fruits, vegetables and storage organs. If you consider increase in cell number increase you can see in maize root apical meristem, like 17,500 new cells are formed from a single cell. 17,500 new cells are formed for every hour. Okay. This is increase in cell number observed in bacteria, algae and yeast. Then increase in surface area is observed in leaves. Increase in volume you can see it in fruits, storage organs. Increase in cell size, like a single watermelon cell can expand up to 3,50,000 times. A single watermelon cell can expand up to 3,50,000 times. And here if you see, both increase in cell number and increase in cell size is clearly mentioned out in your text. And increase in length you can see, increase in length is also measured through pollen tube increase, pollen tube length increase. Increase in length by pollen tube increase. Fine, clear till here? Any doubt anywhere? And then phases of growth. About phases of growth we have already learnt in morphology, where we are addressing the regions of the root. Here also the characteristics of the cell will be the same in phases of growth. Look into the meristematic phase. How the meristematic phase, cells will be in the meristematic phase. Cells in the meristematic phase, cells will have thin cellulosic cell wall, and they have abundant plasmodesmatal connections in between the cell. Okay, they have plasmodesmatal connections in between the cells. Plasmodesmatal connections for easy translocation of minerals and nutrients. And then next is large nucleus is present inside the cell. Inside nucleus, inside the cell, nucleus size is large sized nucleus. Is present inside the cell. So, cells will be having thin cellulosic cell wall and they will be having abundant plasmodesmatal connections, large sized nucleus is present and high rate of respiration is present because of active cell division. Because of active cell division in the zone of meristematic activity, there is increase in rate of respiration. In this cell. These are the characteristic features of the cells which are present in the meristematic zone. And three major changes that happen when the cells are present in the elongation zone. Cells will increase the vascularization, okay. Increase in vacuolation and secondary cell wall depositions will happen. Cell wall depositions. As vacuole size inside the cell increases, this will also lead to increase in cell size or cellular enlargement also you can say it as. or cell enlargement. Fine, right? And in the maturation zone, if you observe maturation zone, fine. Here in the maturation zone, cells undergo differentiation, both structural and physiological differentiation. Both structural and physiological differentiation. Clear? Any doubts anywhere? Clear till here? And then, yeah, look at the growth rate in the plants. In plants, if you see growth rate, what is growth rate? Increase in growth per unit time. Okay, growth rate is nothing but increase in growth, increase in length or increase in number of cells. You can consider increase in growth per unit time. Growth rates are measured in two quantities. One is arithmetic growth rate and second will be geometric growth rate. Arithmetic growth rate is observed in basic plant cells. You can take the example of root apical meristem. Okay, example, best example you can take is root apical meristem or growth in the root region of the plant, wherein a single cell undergoing mitotic division forming out two daughter cells, of which one cell will be meristematic and other cell gets permanent. Again, when the meristematic cell undergoes mitotic division, one cell will be meristematic and the other cell will get permanent. Fine. Now, here you see the geometric growth rate, wherein this is observed mostly in the bacterial culture. In bacterial culture, where a single cell dividing into two cells. Each cell is meristematic and each cell will further undergo the cell division. Thereby, there will be huge increase in cell number. When the graph of arithmetic growth rate is plotted on the x-axis, time is taken both for arithmetic growth and geometric growth. On x-axis, time is taken. On y-axis in arithmetic growth, height of the plant is considered, and here on the y-axis, size or weight of the organ is considered. Fine, right? So, here if you observe the growth rate, you can find linear growth rate completely. Okay, there is a linear growth rate. Fine. Linear growth rate is observed in the height of the plant, and it is calculated as total length is equal to initial length plus growth rate. Okay, rate of growth and T will be time. And here the formula to calculate the geometric growth rate will be final weight is equal to initial weight into logarithm of the growth rate, ERT. Okay, log of growth rate is taken. Fine, right? So, these are the formulae to calculate both arithmetic growth rate and geometric growth rate. And here you can see increase in growth per unit time in root apical meristem, and this is how the growth is being happening both in the arithmetic growth rate and in the geometric growth rate. Clear till here? Any doubt anywhere, ma? Now, yeah, here we go with first. If you see the zygote which is dividing, for zygote immediately after the formation of a zygote, first form of growth you will observe is geometric phase of growth, where the single cell will completely divide and each cell which is formed at the end cell division will start dividing, and this forms a complete embryo. Once the embryo is formed, then few cells starts differentiating for the organ formation, At the time of differentiation, you can see the arithmetic phase of growth, means few cells will lose the capacity of division and they will start the capacity of differentiation. Clear, right, ma? Zygote, first it will undergo geometric growth rate and then you can see arithmetic phase of growth. Here you see measurement of growth. You can observe growth is measured quantitatively in two ways. Okay. Quantitative measurement of growth. Quantitative measurement of growth. We have two types of measurements in a quantitative way. First is absolute growth rate and second will be relative growth rate. Remember, first will be absolute growth rate and second is relative growth rate. Fine, right? Absolute growth rate and relative growth rate. Here we go first with absolute growth rate and then you have relative growth rate. How you will calculate the absolute growth rate? Here if you see in image 1 and image 2. Image 1, here if you can see the diameter of the leaf, it is considered as A, which is of 5 centimeter square. And here if you see A dash, here you have the length increase is up to 10 centimeter square. So initial size is 5 centimeter square and the final size over here is 10 centimeter square. For first leaf, if you consider the absolute growth rate, the initial size, absolute growth rate formula will be final size minus initial size. If you consider relative growth, it is calculated as final size minus initial size by initial size into 100. Fine, right now. Absolute growth rate, if you see, it is from final size minus initial size. Relative growth rate, if you observe, final size minus initial size by initial size into 100. Okay. So, here you observe first absolute growth rate of leaf 1, first leaf. Look at here carefully, absolute growth rate and relative growth rate. First, final size 10 centimeter square minus initial size 5. It is 5 centimeter square. Here for leaf B, if you calculate the absolute growth rate, it will be final size 55 minus initial size by initial size into 100. Okay. So, if you calculate this, it is 10 centimeter square. Clear, right, ma? Sorry, this is for relative growth rate, right? For absolute growth rate only you see, it is 55 minus 50, the answer will be 5 centimeter square. Relative growth rate here, final size minus initial size by initial size into 100. If you see, it is 10 centimeter square, right? And here if you observe 10 minus 5 by 5 into 100, it is 100 centimeter square. Clear with absolute growth rate and relative growth rate? Any doubt anywhere, ma? Is this clear? Absolute growth rate and relative growth rate, very important. Application based questions have been asked quite a times in your previous year questions, both for NEET and EAPCET. If you are having any doubt, you can ask me here itself. Then the concept of differentiation, de-differentiation and redifferentiation. This concept will be asked in connection with anatomy of flowering plants, for which you have to have a clear idea, you should have a clear idea on the anatomy of flowering plants as well. So, here if you consider firstly the meristematic regions of the plant, first meristematic regions. You can see meristematic regions which include shoot apical meristem and root apical meristem. We have two types of meristematic regions. One is promoting apical growth and other is promoting intercalary growth. Shoot apical meristem, root apical meristem and intercalary meristem. These are different types of meristematic zones. Fine. And then you see we have after meristematic regions, second is permanent tissues which are formed. Okay. As they are following arithmetic growth, few cells will start differentiating. So, first differentiated regions and differentiating tissues you observe. Let us see differentiated regions and tissues. Differentiated tissues will be both primary and both simple permanent tissues and complex permanent tissues. Fine, right? Simple permanent tissues and complex permanent tissues. If you see simple permanent tissues, you can consider parenchyma, collenchyma and sclerenchyma. Complex permanent tissues like xylem and phloem, all these are differentiated tissues only. But if you observe differentiated regions you can see cortex, epidermis, endodermis, pericycle, vascular bundles, that is both primary xylem and primary phloem. Last, pith or medulla. These are the differentiated regions. And next, if you consider, third is de-differentiated tissues. De-differentiated. Few cells in the cortex region, they will gain the capacity of cell division again, both in the cortex region and in the stellar region, to conduct both stellar secondary growth and extrastellar secondary growth. For this, what happens? The tissue which is giving rise to the meristematic activity in the cortex region, the tissue which is giving rise to the meristematic activity again is addressed as cork cambium, and in the stellar region the tissue which is giving rise to the meristematic activity is addressed as vascular cambium. Okay? The tissue which is conducting the cell division activity again in the cortex region is addressed as cork cambium, and the tissue which is conducting the the division growth again in the stellar zone is addressed as vascular cambium. So, here the de-differentiated tissues will be vascular cambium and cork cambium. Vascular cambium in stellar region and cork cambium in extrastellar region. Stellar region and extrastellar region. Clear, right? And after this, if you observe, redifferentiated tissues. Redifferentiated tissues are those tissues which are formed by the meristematic activity of cork cambium and vascular cambium, both in the stellar region and in the extrastellar region. That is, you can consider cortical region and vascular region. In both the regions, the new tissues, the permanent tissues which are formed by the activity of vascular cambium and cork cambium are addressed as redifferentiated tissues. And the regions include secondary xylem and secondary phloem in the vascular or stellar region. You can also observe secondary pith or medulla and secondary medullary rays. And in the cortical region, if you see, you can observe secondary cortex and secondary cortex and cork cells or phellum. So, the concept of differentiation, dedifferentiation and redifferentiation is asked multiple times in your objective exams in connection with the chapter anatomy of flowering plants. So, highlight this and learn, okay. Mark this, highlight this and learn. Fine. And next, development. Here you see, first meristematic cell we have observed. This meristematic cell will undergo plasmatic growth, okay, that is increase in cell size and cell number, and it forms few permanent tissues. Permanent tissues will be formed after differentiation, and this is by elongation and maturation. First, plasmatic growth, that is increase in the meristematic cells by cell division, and then increase in the cells, increase in vacuolation, after which both structural and physiological differentiation will take place and it will form the mature cells. These mature cells will undergo senescence or death. Fine, right? This is how the complete development process or the sequence will happen. Okay. Development in simple terms you can learn the combination of growth and differentiation. In simple terms it is growth along with differentiation followed by senescence is known as development. The sequential events or sequential events that happen, first growth followed by differentiation and senescence, is addressed as what? Development. Okay, that is sum total of all events, growth, differentiation and senescence. And here comes the concept of plasticity. What is plasticity? Plasticity is nothing but same plant giving different growth patterns. Okay, same plant showing different growth patterns and different growth structures. It is forming different growth structures both in response to environment or in the response to phases of plant life is observed as plasticity. Okay. Different structures developed due to diverse growth patterns which occur in response to either phases of growth or in response to different habitats the plant is living. Same plant growing in different habitats will show different structures in response to different growth patterns. That is known as plasticity. So, first if you see the first response to different phases of life or phases of growth. If you observe coriander in your daily life, coriander leaves, the juvenile phased coriander leaves will be of different structure, and adult coriander leaves you can see they are of different structure. One phenomena or one example of plasticity will be heterophily. Hetero, different, phily, leaf structures. Different leaf structures are observed by the same plant in two different phases, one in juvenile phase and one in adult phase. What is this, ma? Heterophily. It is the response of different phases of growth. Fine, right. So, here if you observe the examples include cotton, coriander and larkspur. And the same phenomena which is addressed as habitat heterophily. Same species growing in two different regions, one in the terrestrial habitat and two in the water habitat, and this shows different leaf patterns. This is addressed as habitual heterophily. Or heterophily based on habitat. Example, buttercup. Fine, clear till here? Any doubt anywhere? Here the examples, if you consider both for phases of growth and heterophily based on habitat, both are important. Examples are very important. Here it is cotton, coriander, larkspur, and here it will be buttercup. Fine, right. And if you observe growth factors, growth of plant is affected by many factors, both internal growth factors and external growth factors. If you observe internal growth factors, it includes intracellular genetic factors and intercellular factors like chemicals which are plant growth promoters. Now, we are going to learn about plant growth promoters in detail. And extrinsic growth, extrinsic factors which limit the growth are light, temperature, water, oxygen and nutrition, about which we have learnt in detail in photosynthesis in higher plants. So, now when we consider the intrinsic or internal factors which affecting the growth of the plant, intracellular factors, those are genetic factors, and intercellular plant growth regulators. Now, we are landing on the concept of plant growth regulators. Plant growth regulators are of two types. Plant growth regulators are also known as phytohormones. These are known as phytohormones. These are small simple chemical constituents which are synthesized or stimulated in very small quantities, and they show diverse activities in different regions of the plant. That is about phytohormones. We have two types of phytohormones. One is plant growth promoters and second type is plant growth inhibitors. We have both. Plant growth promoters and second will be plant growth inhibitors. growth promoters include auxins, cytokinins, and gibberellins. And plant growth inhibitors is abscisic acid, ABA, and ethylene. If you see ethylene, it will have dual functions, both as plant growth promoters and plant growth inhibitors. So, for today's session, what all we have learnt, ma? First, starting from seed germination on two types of seed germination, both epigeal and hypogeal seed germination, followed by viviparous germination observed in mangroves which are growing in the plants which are close to the seashore, followed by open form of growth which is because of lateral or secondary meristems, and then growth is measurable on many parameters in plants we have observed, of which the most important one is increase in cell size, increase in number, and increase in the length, and its examples. Fine, right? And then phases of growth, meristematic, elongation phase and differentiation phase, after which we have learnt growth rates, both arithmetic growth rate and geometric growth rate, including graphs and examples, followed by zygote, which is showing firstly geometric phase of growth, followed by cell differentiation, which is arithmetic phase. Then measurement of growth, quantitative measurement of growth, both absolute growth rate and relative growth rate, followed by differentiation, de-differentiation and redifferentiation concept, and then development, which is the sum total of events from growth, differentiation and senescence, and then about the concept of plasticity and heterophily. Lastly, about the factors affecting growth, and one of the internal factor is plant growth regulators or phytohormones. What are phytohormones we have learnt, and what are plant growth promoters and inhibitors we have understood. In our next class, we are going to sum up or complete plant growth and development, where clear discussion on plant growth regulators will happen, post which we will exercise the objective from complete plant physiology. EAPCET previous year questions from complete plant physiology we are going to solve. So, come prepared for the next class, where we will have a mass objective session. Fine, right?